Hello, today we are talking about influence of mechanical noise inside a scanning electron microscope. This is our group member is Liu Jiaxun, Zhen Minlun, and Xu Junxiang. This page is the source of this article. Okay, so next I will introduce the scanning electron microscopes. Scanning electron microscopes are high-resolution imaging tools that use an electron beam interacting with the sample surface to generate images. They are primarily used for observing surface morphology and analyze sample composition. I will state about the principle. The E-beam scans the sample surface, and detectors collect secondary and backscatter electron signal to form high-resolution images. Next is the advantages: high resolution, large depths of field, and capability for micro-level composition analysis. Next will be the suitable samples, include metals, polymers, ceramics, and biological materials, though non-conductive samples often require coating. Why use scanning electron microscope instead of optical microscope for observing microstructures? They have 6 opinions in this paper. First is resolution limitations. Second is depth of field. Third is surface detail presentation. Fourth is magnification. Fifth is sample characteristics and analytical capabilities. Sixth is application difference. Okay, I will using SEM to compare with OM. So the first is resolution limitations. OM are limited by the wavelength of light, with a theoretical resolution of approximately 200 to 215 nanometers. This makes it difficult to observe nanometer-scale structures or smaller details. And SEM use an electron beam with a much shorter wavelength, achieving resolution as fine as 1 nanometer or even lower, making them ideal for studying tiny structures. The second thing is depth of field. ON has a limited depth of field, offering a narrow focus range that makes it challenging to clearly observe multiple layered structures. And SEM provides a much greater depth of field, allowing for clear visualization of surface structures with depth, making it suitable for 3-dimensional morphology observations. And next will be surface detail presentation. OM struggles to display fine surface details and often lacks sufficient concentrate to reveal intricate structures. And SEM, utilizing secondary electron and backscattered electron detectors, provides sharp surface details and high concentrate, revealing even subtle features. And the fourth is magnification. OM has a maximum magnification of around 1000 times, which is insufficient for examining nanometer-scale structures. SEM offers magnifications up to several 100,000 times, enabling detailed observations of microscopic features. And the 5 is sample characteristics and analytical capabilities. OM cannot directly analyze a sample's composition. It is limited to optical observations. SEM can be equipped with energy dispersive X-ray spectroscopy to analyze a sample's composition, providing both structural and chemical information. And the final is application difference. OM is suitable for quick, Sample inspections of larger scale samples or those with relative raw surface. SEM is better for cases requiring high-resolution observation and detailed structural analysis, such as biological tissues, nanomaterials, or electronic components. So others working in micro-manipulation in SEMs resort to embedded sensors and imaging feedback to perform the tasks, while indicators can be statically positioned with accuracy in an order of a few nanometers through the use of filters, i.e., averaging algorithms, statistical methods, etc. These methods are not adapted for dynamic performance, as they require longer multiple acquisitions. And by the way, the author is studying a SEM with a cantilever and used for nano-level fine operation. So the author guesses what factor will reduce accuracy: acoustic wave, SEM noise, position table vibration, ground vibration, vacuum pump vibration. This figure is the reason for the author's guess.
Hi, it's Liu Jiaxun. The student number is number 1. Now I'll introduce experiment method and equipment. All the experiments were done under high vacuum, which is typical SEM environments. Besides, the author used 4 cantilever in order to reduce random measurements noise. For each cantilever, 20 measurements were performed. Then the author averaged the result. Here is the vibrometer what the author used in their experiments. It's called laser Doppler vibrometer. It's a high precision, non-contact instrument used to measure the vibration of an object's surface. Basically, a laser beam is launched to the vibrating surface. When the surface vibrates, its motion causes slight shift in the frequency of the reflex laser light and leads to Doppler shift. This signal is detected by vibrometer and processed to exact details. Here's the graph, shows the real equipment. The author fixed the vibrometer to the SEM door at a 45-degree angle so that the vibrometer would not block the electron gun. So you can see the white part and say SIOS. That is the vibrometer what the author used in their experiment. Let's move to the results of the initial test. You can see there are several abnormal vibrations. Later, my teammates will introduce how the author tells the vibration source.
And the by changing the position of the vibrometer, the author observed that 2 of these peaks shoot to 82.3 Hz and 122.5 Hz, while the other peaks remain unchanged. This confirmed that these 2 peaks were caused by the vibrometer positioning. Then the author added weight to the positioning table. The frequency shifts again when weights were applied. Therefore, the author attributes these 2 peaks to the positioning of vibrometer. To identify the source of the remaining peaks, the author conducted the following investigations. First, the author turned the vacuum pump on and off. In the resulting displacement, the blue line represents the pump on state, and the red light presents the palm of the. Compare the two. The author found that strong vibrations occurs in a range of 660 Hz to 720 Hz when the palm was running. Next, the author turned the SEM on and off and analyzed the displacement. The author discovered that around 955 Hz, there was significant interference caused by electronic noise, which resonates with the original vibrations, leading to amplified oscillations. Finally, the author identified the interference source for all 6 major peaks. However, when the sound matches a reasonable frequency, vibration will amplify significantly even at a lower sound level of 64 decibels.
Hi, it's me again. I'll talk about the conclusion. First, the author shows the most important disturbance that can influence manipulation in SEM environments. Second, this analysis of dynamic behavior within the chamber provides information for real-time dynamic noise rejection, which is important to achieving fast and precise manipulation and positioning. Third, the author also demonstrated that acoustic disturbance can enter through the microscope's walls, impacting accuracy inside the vacuum chamber. Finally, this study shows that the importance of take both environmental and internal disturbance seriously to meet the extreme precision requirements for SEM-based tasks, which, with careful consideration of these factors, we can improve the reliability and accuracy of operations in SEM. And that's all of our presentation. Thanks.